I'm Roger Alexander, the Scientific Outreach Coordinator for the Extracellular RNA Communication Consortium. Today's speaker is Michael Segel from the Zhang lab at MIT. He's the first author of a recent science paper on looking for retroviral-derived proteins in the human genome, which have been functionalized, and they specifically studied one called PEG-10, which encapsulates its own mRNA and traffics it to other cells. And they have started to work engineering that as a vector for other RNAs, as a therapeutic vector. So, Michael, we're really looking forward to hearing you talk. Just quickly, if you have questions during the talk, put them in the chat. I'll ask those first at the end before opening it up to everybody.
Well, hi everybody, I'm Michael Segel. I'm a postdoc in Feng Zhang's group, as Roger just said, here at the Broad Institute and at MIT. And today I'll be talking about our work looking for retrovirus-like proteins within the mammalian genomes to develop new RNA delivery vectors. So just to give you a brief background about where my academic training was and how this project really began. I did my graduate work in neuroscience at the University of Cambridge with my major focus on how the many cells within the central nervous system communicate and coordinate development. So this picture here is not my work, but instead a really useful EM-based reconstruction of the cells of the central nervous system, which really showcases the sheer diversity and cell density of the central nervous system. So with my background lens of neuroscience and biology, I was particularly attuned to the vein of literature that came out a few years ago from Travis Thompson's group and Jason Shepard's group, looking at this two analogs of this protein called ARC, which they found are capable of forming capsid-like structures and are able to transfer nucleic acids from one cell to another cell in both the central and peripheral nervous systems. Now, both of these analogs of ARC, both the Drosophila one and the mammalian one, are retroelement derived, meaning that the proteins evolved from some ancient retrointegration event. So retroelements are particularly interesting because upon entering into a cell, they're able to reverse transcribe their RNA genome into double-stranded DNA and then incorporate that double-stranded DNA into the host cell's genome. And once in the host cell's genome, they're able to coax the host cell machinery to produce more retroelement RNA transcripts, produce more retroelement proteins, and then subsequently coax or trick the cell into mediating additional retrointegration events. Now, if these retrointegration events occur in a germline cell, like a sperm or an egg, then these retrointegrations are actually passed down from one generation to the next, or in some cases, even from one species to the next throughout evolution. This has actually happened quite frequently throughout evolution, and here is just on the top, you can see a nice schematic of the canonical or archetypal retroelement. And on the bottom, you can see that throughout the genome, the human genome, there's a smattering of different retroelement-derived sequences. So some of them are fully intact and are our enemies, and we try very hard to keep them from replicating. But other ones, like these bottom panel of proteins, which encompasses not so many, but a substantial number of our own coding sequences in our genome, are actually domesticated or retooled retroelement-derived sequences that we've acquired from retrointegration events, and our ancestral cells have figured out how to retool certain useful aspects of them to perform key cellular roles. And so just to go through some background on this, there's on top, you can see, again, the schematic of the retroelement. So canonically, they have these three domains, the GAG, POL, and ONV domain. And so the GAG domain is the key structural component of the retroelement. And so when you think of a retrovirus, this is the beautiful spherical capsid that you see. And then you also have the POL domain. So this is the domain that mediates the reverse transcription of the retroelement RNA genome and integration into the host. And then you have the ONV domain, which is unique to retroviruses, which enables these extracellular particles or viruses to enter into host cells and thereby deliver the retroelement payload. So all three of these domains have actually been retooled by mammalian cells to enable key cellular functions. So the ONV protein has been retooled, or certain ONV proteins have been retooled by all placental animals to enable the fusing and the evolution of a fused barrier placenta. And in mouse, when you delete these proteins, the fetal mice aren't able to form protective placentas, and this means that the pregnancy is terminated. So this is an essential protein for all mammalian development. Another protein which people don't generally think of as derived from a retroelement is telomerase, so the famous protein that maintains the ends of chromatin. And this is derived from a retroelement reverse transcriptase protein. And then, as I've already talked about, ARC has long been known to have key roles in central nervous system functions and memory consolidation. And this was known well before it was even concretely established that this was indeed a retroelement-derived protein. And so with this wealth of literature at the get-go from when I started my own project, I really started by trying to think, well, are there any other retroelement-derived proteins like ARC that are capable of transferring nucleic acid encoded information from one cell to another cell? And then the corollary question, because of my neuroscience background, is what role would the biology of these such a system? What role would such a system have in the biology of the central nervous system? And so, with these basic questions at hand, I really went back and thought, well, we had to go back and hypothesize, well, at minimum, if such a system of intercellular RNA communication were to exist, we would really think that at minimum it would contain the structural component of a retroelement, so this capsid domain. And so with this basic hypothesis, we reached out to our longtime collaborators in this lab, Eugene Koonin at NCBI, and he really helped us look across all mammalian genomes for any proteins that contained a capsid-like domain in it. And so we started out with collating hundreds of capsid-like domains that are scattered across mammalian genomes. But we really wanted to further hone what we were looking for, because otherwise it was just too many proteins to begin to even think about. And so we started by thinking, well, one, are there capsid domains? This is what I just said. And two, are they conserved across mammals? Because if they're conserved, then maybe these really are retooled or important proteins for some new mode of intercellular communication. And the third search parameter we had is there any RNA transcription of these identified proteins in the adult human? And this really, again, would bolster the second genome mining parameter, and really suggest that these proteins are actually important and serve some role in the biology of the adult human. And so with these three search parameters, we really narrowed down to just 10 capsid domain containing proteins. And here you can just see a schematic of all the proteins that we'd identified. So as you can see from this orange box, all of these proteins have a predicted capsid-like domain. And from this yellow diagram box here, you can see that many of these proteins also have RNA binding proteins, the RNA binding motif of a retroelement called the nucleocapsid domain. But other ones have a different smattering of these retroelement-derived proteins. So some of them have this nucleocapsid domain, some of them have protease domains, whereas others, like these last two, have a reverse transcriptase-like domain. So we've identified these proteins computationally. But our next question was, well, do any of these proteins actually form capsids? And can we empirically show this? So we, again, borrowed heavily from the previous papers on ARC and really just tried put these proteins in a very unnatural context. So we overexpressed each of these proteins in E. coli, and then did really stringently purify these proteins using column purification and size exclusion. And we just let these monomeric each of the monomers of these proteins oligomerize with themselves overnight to see if they could form some sort of capsid-like structure, and then we just did electron microscopy on them. And what we found is that out of the 10 proteins that we initially looked at, 9 of them formed these beautiful and strange circular capsid-like structures, just ex vivo, so on their own. And you can see on the left these nice negative stain electron micrographs, and on the right you can see these more zoomed in, higher resolution cryo electron micrographs. And so we were very excited to see that many of these proteins that we identified computationally actually do form capsids as we predicted. But our next question was, well, it's great that these proteins form capsids, but are any of these proteins released by mammalian cells as are bona fide retroviruses? So are any of them found in the extracellular fraction in cell culture? And so to do this, we did another very simple experiment, which is we just epitope tagged N-terminally and C-terminally epitope tagged each of these capsid domain containing proteins with an HA epitope tag, and we heterologously overexpressed each of these proteins in HEC293 FT cells. And we harvested the self-free fraction of these HEC293 cells, and we purified any extracellular vesicles via ultracentrifugation. And when we just blotted did Western blots on these extracellular fractions for this HA epitope. And what we found is that of the 10 original proteins that we were interested in, at least 4 of them, using this very rough technique for determining it, at least 4 of them were heavily secreted proteins as detected by Western blot. So this was again, very compelling, because not only do these proteins form capsids, but at least some of them also are secreted proteins, as are real retroviruses. So we next wanted to see, well, in real tissue, are any of these proteins found in the self-free fraction? And of all the ones we looked at, really only one of them were we able to find abundantly in the self-free fraction of mouse blood serum. So this is ultracentrifuged mouse blood serum. And we did Western blots using commercially available antibodies. And we see that, yes, this one protein, PEG10, which is one of the ones we had previously identified, does seem to appear abundant in both blood serum and in brain. So it's great that these proteins form capsids and are secreted proteins, but are these proteins actually being secreted as capsids? Because a lot of proteins are secreted proteins. Insulin, for instance, is a great secreted protein, but it doesn't get secreted as a capsid. So we next reached out to our friends at Harvard Medical School to see if these proteins are actually being secreted as capsids. And what they did is they did these nice semi-thin sections of cells that were heterologously overexpressing these different proteins. And they did semi-thin sectioning and immunocold labeling. And what we see is that we can actually see these particles being released in what appears to be some sort of multivesicular body. But they are indeed being released, what appears from our rough analysis, as capsids, as lipid-bounded capsids. So this is really our first evidence that these proteins are actually being released by mammalian cells as capsids. So our next question is, this is great that these proteins are released by capsids, but are they actually encapsulating any sort of nucleic acid encoded information as they're being released from cells? And so to ask that question, we had to kind of go back to the drawing board and do a little bit of brainstorming, because as many of you guys could appreciate, it's very hard to study extracellular RNAs, because oftentimes the RNAs that you detect via next generation sequencing or qPCR even are often contaminating RNAs that come from either cell lysis or just cell debris, or just from the transfection reagent. If you're putting in a plasmid into a cell, you oftentimes just detect that plasmid when you do any sort of next generation sequencing or analysis of the RNA in this extracellular fraction. So this is a long-winded way of saying, so to overcome this, we use this technique called CRISPR activation, where we don't just overexpress a gene of interest, but instead we use the CRISPR machinery to bring transcription activation components to a specific genomic locus, and thereby activate transcription and translation of a given protein. And so this roundabout way allowed us to assay whether or not endogenously transcribed and translated capsid domain containing proteins are actually encapsulating mRNA and releasing them from cells. So we used this CRISPR activation approach. We harvested the cell-free fraction we nuclease treated the self-free fraction, and we did total RNA sequencing on both the whole cell and the cell-free fraction. And what we found is that after looking at a number of the different capsid domain containing proteins with this experimental pipeline, we found that the only transcript, really, the only mRNA transcript, I should say, that was significantly enriched for in the cell-free fraction was the transcript for PEG10. And again, this is that protein that I've been talking about in the last couple of slides. And so PEG10 transcript, when you transcriptionally activate the PEG10 mRNA transcription and its subsequent translation, you actually detect an enrichment of PEG10 mRNA transcript in the cell-free fraction. And you can see these nice read histograms here. And this is, again, a nice, we were very much inspired by previous work from Travis Thompson's group's paper, which has a similar read histogram in their paper. And so we found that PEG10 is not only a secreted protein, that it's secreted as a capsid, but that it's also, when it's secreted as a capsid, it appears to be secreting its own mRNA. So this was really the point at which we became very excited about PEG10 and wanted to pursue it a bit further. And so let's just go back to the drawing board. PEG10 is a very long capsid-forming polypeptide, which has been shown previously to be required for embryonic development. So if you knock it out, it's embryonic lethal at day 8, and we recapitulated those findings. And yes, it is indeed embryonic lethal. And it's a very long polypeptide with two overlapping reading frames, reading frame 1 or RF1 and reading frame 2 or RF2. And so this, as someone from the outside, this really begins to look very similar to something like a real bona fide retroviruses, which similarly has two open reading frames. So the first open reading frame encodes the capsid domain, which is what we mined for, but also this RNA binding nucleocapsid domain. In the second reading frame, you can see that it from the schematic, that it also encodes a protease domain and a heavily truncated and catalytically inactivated reverse transcriptase domain. So this is not a real reverse transcriptase protein. This is just some reconfiguration that some ancestral mammalian cell has repurposed or domesticated for some unknown function. And we did some analysis of how the PEG10 polypeptide gets processed within a cell. And what you can see from this simple schematic is that the protease domain is actually responsible for the processing and the cleavage of this long polypeptide into each of its predicted functional domains. So the capsid domain gets proteolytically processed away from the nucleocapsid domain, and the reverse transcriptase domain also gets cleaved and proteolytically processed. In much the same way that a maturing HIV retrovirus gets processed down into each of its predicted respective functional domains. And so our first question is, well, there's two putative RNA binding motifs of the PEG10 polypeptide, the nucleocapsid domain and the reverse transcriptase-like domain. So are either of those two domains responsible for this RNA binding encapsulation and secretion of the PEG10 mRNA transcript from a cell? And so what we did is we mutated either the nucleocapsid domain or the reverse transcriptase-like domain and just heterologously overexpressed these different mutants of PEG10 in HEC293 cells, in mouse neural 2A cells, and then just again harvested the cell-free fraction from these cells and did qPCR for the PEG10 mRNA transcript. And what we found is that, and I don't show this here, but each of these different mutants of PEG10 is secreted as a protein, but when you delete the nucleocapsid domain, you no longer detect significant levels of PEG10 mRNA transcript in the cell-free fraction. So this really suggests that the predicted RNA binding nucleocapsid domain is responsible for the binding to encapsulation and secretion of the PEG10 mRNA transcript itself. And so our next question was, well, how does the PEG10 protein actually recognize, bind to, encapsulate, and secrete the PEG10 mRNA transcript? Is there any sort of sequence motif that allows it to identify its own transcript to allow for the specific secretion of the PEG10 mRNA? And so to answer this question, again, we really had to go back to the drawing board and think about what is the cleanest way that we could do this. So unfortunately, there's not very clean or efficient and commercially available antibodies against PEG10. So we had to actually generate mouse strains in which we've knocked in the HA epitope into the N term or C term of the endogenous PEG10 locus in embryonic mice. And so we generated homozygous mice with this HA knock-in, and we let these mice develop into an adult. And then we harvested the cortex of these transgenic mouse strains. We're able to purify PEG10 protein RNA complexes by using an antibody against HA, which is a very well-established antibody. We UV cross-linked any protein RNA interactions. We fractionated it and did some RNase footprinting, and then did next-generation sequencing. And with this approach, we were able to generate a very clean readout of how the PEG10 protein binds to its own PEG10 mRNA transcript. And so here is just another read histogram of the reads we got back from these RNA immunoprecipitation experiments. And what we found is that the PEG10 protein really, at least in the cortex of a mouse, really likes to bind to the non-coding regions of the 5' untranslated region and the 3' untranslated region of the PEG10 mRNA transcript. So with this RNA binding information, we next hypothesized that maybe these non-coding sequences constituted some sort of signaling packaging sequence that we could maybe flank onto a gene of interest, maybe some sort of therapeutically relevant gene, and tag that gene of interest to be encapsulated by the PEG10 capsids, have that gene of interest get secreted, and then perhaps even have that gene of interest get delivered to a target cell to develop some sort of new RNA delivery vector. And so this is what we tried. So in HAC293 cells, we transfected in 3 different plasmids. We transfected in the PEG10 plasmid. We also transfected in this cargo RNA, which is what we call a gene of interest flanked by these non-coding packaging sequences. And then we also hypothesized that if we're generating extracellular capsids, then maybe they need to be functionalized with some sort of cell entry mechanism. And so here we also transfected in a fusogenic protein. And in this case, we used the viral-derived fusogen called VSVG. And because that's a very well-established broad tropism fusogen, which can get extracellular vesicles into a lot of different cell types. So we triple transfected these 3 plasmids. We generated extracellular particles with PEG10. And in our first iteration of this experiment, we tried packaging Cre-recombinase into Cre-recombinase mRNA into these virus-like particles, or VLPs. And then we purified these virus-like particles via ultracentrifugation and various gradient fractionations to get really pure preps of these extracellular virus-like particles. And here is just an example of one of the fractionations that we get. This is a Western blot of following an iodixonol gradient, which is a way of purifying extracellular particles of different sizes. And then we took the different fractions of this iodixonol gradient here, and we did just electron microscopy, negative stain electron microscopy. And what you can see is in this fraction here, which co-elutes with this with the TSG101 fraction, which is a canonical marker of exosomes, that we see that we have these very relatively homogeneous extracellular particles. And intriguingly, they look very not super different by our untrained eyes from previously published extracellular particles that were generated from cells that were transfected with the components for HIV. So it really seems they look by our untrained eyes to be quite similar to real bona fide retrovirus particles. And here's just a more zoomed in version. And I just really like this picture because it really allows us to begin to see what these extracellular particles look like. And so you can see what maybe could be some sort of capsid-like structure surrounded by what appears to be a lipid bilayer. And so with these purified extracellular particles containing Cre-mRNA, we then put these extra purified particles onto recipients' LOXP RFP reporter cells. And so in this initial experiment, we learned three very key things. One is that we are able to functionally deliver mRNA to recipient cells with the PEG-10 system. We learned that this process really depended on the presence of PEG-10. This wasn't just spurious contamination of some transfection reagent transferring Cre from one cell to another. And the second thing we learned is that this process of functional transfer wholly depended on the presence of these non-coding packaging sequences that we identified from the CLIP experiment. And so without these non-coding packaging sequences, we really don't see the functional transfer of Cre mRNA from one cell to another cell. And the third really important thing that we learned from these experiments is this whole process wholly depended on the presence of this fusogenic protein, PSVG. So these extracellular particles that we generated really needed to be functionalized with a fusogenic cell entry mechanism to allow for the functional delivery of these cargo Cre or UTR-flanked Cre mRNAs. So we next really needed to show definitively that we're actually transferring Cre mRNA from one cell to another and not just Cre protein or some sort of cell debris, which is, as you can all appreciate, is a very difficult thing in the extracellular vesicle field. And so what we did is a number of different next generation sequencing experiments and just qPCRs of the self-free fraction. And what we found is that while we're not generating RNA titers equivalent to that of a lentivirus, we are getting large levels of Cre mRNA in the cell-free fraction from cells transfected with the PEG-10 plasmids. And then in addition, we show via Western blot that we're not just actually transferring Cre protein from one cell to another cell. So when we Western blot the cell-free fraction, the ultracentrifuged purified VLPs, we don't really detect any Cre protein, though we are able to detect the PEG-10. So we have the PEG-10 particles, and we detect Cre mRNA, but we're not detecting Cre protein. So this is pretty good evidence that we're actually transferring Cre mRNA, not protein, from one cell to another. So this is great. We seem to have a system working for mouse, but our next question to really concretely show that the system indeed works as we think it does is to translate the findings from mouse into the human ortholog of PEG-10. And so we pretty much just wanted to recapitulate the same findings from mouse in a human system. And so as you can see here from these conservation tracks from the genome browser, that the PEG-10 is actually very highly conserved between species. So here you can see the human PEG-10, even the intronic region is conserved between mouse and human. And the 5' and 3' UTRs of PEG-10 are also similarly highly conserved between mouse and human. And so this gave us some confidence that the human system would work very similarly to the mouse one. So maybe the non-coding UTRs are similarly important for the functional transfer of mRNA. So just again, very basically, a very basic experiment is we just heterologously overexpressed the human PEG-10 and then blotted the cell-free fraction to see if the human PEG-10 is similarly secreted as is the mouse PEG-10. And here's what we found. This is the whole cell. You can see this nice processed polypeptide into the different predicted functional domains. And then in the cell-free fraction, you can see again, different processed versions of the PEG-10 polypeptide. And so human PEG-10 is similarly secreted as is the mouse PEG-10. And so now that we have a system working for both mouse and human, we wanted to hone in and really try to understand which regions of these non-coding packaging sequences were really important for the functional transfer of Cre mRNA from one cell to another cell. If we go back just one slide, oh, sorry. You can see that these non-coding packaging sequences that we are flanking Cre with are very large. They're at least 3 kb in both the mouse and the human. So we wanted to zoom in a bit more and get a better understanding of which sequences were important. And so what we did is we just took 500 base pair bins of the 5' and 3' untranslated regions for both human and a mouse PEG-10. And we just walked along these non-coding sequences and really tried to understand if we could just shorten these non-coding sequences to identify the precise regions that were responsible for the functional transfer. And what we found is for both the human and the mouse orthologs of PEG-10 that it was really just the proximal 500 base pairs of the 3' UTR that were alone necessary for the functional transfer of Cre mRNA. And if you overexpress the latter, Cre mRNA is tagged with these latter sequences of the 3' UTR, you really lose most of the functional transfer. And so all we need is these proximal 3' UTI. So this is a much more compact system now, and with a lot more defined non-coding sequence packaging sequences. So this is one level of optimization that we wanted to do. But the next question we had is, well, we had some preliminary evidence that PEG10 protein really liked to still package its own mRNA as opposed to packaging this Cre mRNA in trans. And one reason we hypothesized that this was the case is if we go back to the CLIP experiments, we see that there's this protein RNA interaction that seems to occur in the middle of the coding sequence here. And so this made us think, well, maybe there are other non-coding sequences in the actual open reading frame of PEG10 that PEG10 likes to bind to and would prefer to bind to than actually these Cre mRNAs that we're providing in trans. So to answer this question, again, we did both for the mouse and the human. We walked along the coding sequence of mouse and human PEG10, and we just swapped out the codons for alternative codon sequences along the transcript. And what we found is that as we different codon usages at different points of the transcripts actually boosted the functional transfer of Cre mRNA from one cell to another. So you can see here for mouse with this one recoded region here, RC4, or with human, which is the recoded RC3, we were able to significantly boost the functional translation of Cre mRNA. And then if we take together these optimized packaging sequences, which I described on the previous slide, and these optimized coding sequences for the PEG10 polypeptide itself, then we were really able to start to get pretty efficient functional transfer of the Cre mRNA from one cell to another cell. So now we're getting, at least from pretty low levels of extracellular virus-like particles, we were able to put on pretty small quantities and get very efficient delivery of Cre mRNA to a target cell. And so the people here at Broad really came up with this term SEND, which is what we describe as this newly optimized RNA delivery vector, which is the selective endogenous encapsulation for cellular delivery. So now we wanted to ask, so that's all well and good that we can deliver Cre mRNA from one cell to another, but what else could we deliver? And so being part of Feng Zhang's group, we were all very interested to see if we could deliver Cas9 to a target cell and mediate some sort of genome editing. So in our first iteration of these experiments, we overexpressed these newly recoded mouse PEG10, a fusogenic protein, VSVG, and then a Cas9 flanked by these optimized non-coding packaging sequences, which we call the cargo Cas9. And we just purified these VLPs, virus-like particles, and put them onto our target cells, which were constitutively already overexpressing a guide RNA targeting the KRAS1 locus. And what we found is, again, we were able to recapitulate our previous results, which is that when we just tried to put on, when we tried to produce VLPs with a Cas9 mRNA that lacked these packaging sequences, we weren't able to deliver we weren't able to deliver Cas9 to the target cells and mediate any sort of genome editing. But when we put on the full-length non-coding packaging signals from the mouse PEG10, we were able to all of a sudden deliver Cas9 mRNA to a target cell. And then with the newly optimized SEND system, which we talked about in the previous slides with these optimized packaging sequences, we were able to get even more genome edits in the recipient cell line. And while we're not quite near the level of gene edits that occur with a lentivirus, so we get huge levels of genome editing with a lentivirus, which is pretty well established, we are now getting significant levels of gene editing in these target cells. And so we next wanted to ask, well, can we combine a guide RNA and a Cas9 mRNA and put it all in the same virus-like particle and put these particles onto naive target cells that don't have a constitutive overexpression of a guide RNA. And indeed, this is what we found. And this was after a number of optimizations that I don't find particularly interesting to go over right now. But essentially, again, we find that these non-coding packaging sequences are important and fully necessary for the functional transfer of Cas9 mRNA from one cell to another. And that when we figured out a way of co-packaging in a guide RNA to also deliver these a guide RNA alongside a Cas9 mRNA into target mouse cells. And excitingly, we were also able to recapitulate the exact same findings with the human system into human cells. So we were able to package a guide RNA targeting a human gene. So this is VEGF-A, and the human packaging sequences flanking Cas9 and efficiently deliver these PEG10 particles and mediate efficient gene editing in these target cells. So this is cool. This shows that this system is pretty modular. We can probably package a number of different mRNAs of varying sizes into these virus-like particles. Cas9 is quite large. It's 4.5 kb. And so we can definitely package something that big. Whether we can go bigger, that's something we're still working out. But one thing that I personally thought was very important for these particles was to generate a fully endogenous delivery vector. So it's all well and good to functionalize these extracellular particles with VSVG, which is again a virus-derived fusogen. But I personally thought I really wanted to make a delivery vector, an RNA delivery vector that was fully endogenous and didn't require a virus-derived protein. And so to do this, we went back to the drawing board again, and we used previously published clip data about PEG10 in the placenta. In the placental trophoblasts. And what we found by reanalyzing this previously published data set is that PEG10 protein really likes to bind to the mRNA transcript encoding this endogenous fusogenic protein called SYN-A. And so SYN-A is, if we go back to one of my initial slides, SYN-A is an endogenous fusogen that actually evolved from an ancient retrovirus integration. And humans also have an analog of this protein, SYN-A. So we hypothesized that maybe we could use this endogenous fusogenic protein to functionalize the PEG10 virus-like particles to enable their entry into target cells and the delivery of an mRNA payload. And so again, we repeated the exact same pipeline as we did previously, where we pseudotyped CRE containing PEG10 VLPs, and we pseudotyped them with VSVG, SYN-A, this protein I was just talking about, or another endogenous fusogenic protein, SYN-B. And what we found is that we were able to pseudotype these PEG10 extracellular particles with SYN-A, which is this endogenous fusogen, but we weren't able to do so with SYN-B, this other one. So we aren't sure why we're not able to do SYN-B, but we think it might have to do with the tropism of SYN-B and whether or not we were using the right cell type, because depending on the tropism of a given fusogen, you really need to ensure that the fusogen actually acts on the cell model that you're working on or the tissue model that you're working on. And so we were able to show that we were able to generate a fully endogenous RNA delivery vector composed of the PEG10 capsid protein and the SYN-A fusogen. And that, again, this whole process of the functional delivery of this CRE mRNA was dependent on these non-coding packaging sequences, which we always like to use as a nice sanity check and control to make sure that we're actually seeing real bona fide functional delivery of mRNA from one cell to another, and not just, again, some sort of like spurious transfection contamination as we generate these virus-like particles. And so just to wrap up what I've been talking about, so we've really begun to develop a modular platform of gene delivery composed only of components that are found within mammalian genomes. So we have, in this first iteration that we talk about in the paper, we have this PEG10 capsid. We find that we're able to encapsulate all these different mRNAs, whether it's mCherry or CRE or Cas9, using the non-coding packaging sequences. And we find that there's a third layer that we need to take into account, which is this fusogenic protein. And there probably are a number of other fusogenic proteins we could try, or maybe they're not even fusogenic proteins. Maybe they're just transmembrane proteins that give real endogenous PEG10 extracellular particles tropism and enable their cellular entry. But here we show that at least this endogenous fusogen SYN-A works and can be used to pseudotype PEG-10 extracellular particles. And together with these three different components, the capsid, the cargo RNA, and the fusogen, we're able to generate efficient RNA delivery vectors composed of largely endogenous proteins. And so the conclusions from this initial study that we've done is that PEG-10 is a capsid-forming protein that is definitely capable of mediating mRNA delivery. It is an endogenous protein, so abundant in humans and most other placental mammals, all other placental mammals. It's very abundant in the adult mouse and human. And because of this, and because it's expressed in thymic epithelial cells, we hope, and this is something that we're working on now, is that it might be able to, these extracellular particles might be able to evade evade provoking an immune response. And the reason why this is interesting, especially at the Broad, where it's people are very interested in the translation of our research. People are very interested in it because redosable mRNA delivery vectors have long been a goal for gene therapy. And as probably most of you are, hopefully all of you are acutely aware, the current RNA delivery vectors are potently immunogenic, which is great for vaccines. But as we begin to look forward for new RNA delivery, RNA therapeutics, which we may want to dose regularly as we dose a traditional pharmaceutical, we might want to develop new RNA delivery vectors that are non-immunogenic, and we could just repeat dose as many times as we want. So what we're really excited about right now is this is again a nice tree diagram from our friends from Eugene Koonen's group at NCBI. And what we can see here is that PEG-10 is actually, the blueprint for the PEG-10 protein is actually scattered across the entire animal kingdom. And so while I'm not saying that PEG-10 is found in fungi, there are PEG-10-like proteins with the similar protein architecture that are really found across all walks of life. And so we're really excited to further explore not just PEG-10, but other similar related proteins, some of which we talk about in our paper, but many of which we have not. And we're really excited to explore further and see what are these proteins doing. Are they involved in the secretion and capsidation of specific mRNAs, as is PEG-10, or do they have other roles, extracellular roles that we haven't yet begun to appreciate? And this is really where I personally see this work going, is just further expanding the world of that Travis Thompson and Jason Shepard really began, and just seeing what else is out there. And so in the short term, the major application questions that we're really pushing on right now is how do we scale these extracellular particles, which is, I think, a problem that many people in the exosome field can appreciate. How do you scale from hex cells extracellular particles? It seems to be a diminishing returns after each ultracentrifugation. And so then also, how should we use these extracellular particles? Can we direct their tropism? Are these particles actually non-immunogenic? That is a very important question that we're actively pursuing right now. And then finally, could we possibly engineer these particles to garner some sort of tropism, or to target them to specific cells? And you can see here just some nice AlphaFold hexamers that we generated a few days ago, and just to see if we were to engineer these proteins. So here's Drosophila arc, and here is Ti3, which is the retro element of which PEG-10 is closest related. And then here's a PEG-10 capsid hexamer, and you can see maybe if we were to want to engineer it, we could tag on something here. I don't know. These are just the sorts of brainstormings that we're having now to really take this project to the next level. And then additionally, some key biological questions we have ongoing is, why would we even have a PEG-10 extracellular particle floating through our blood plasma? What role do these have in normal human physiology, be it in the CNS or elsewhere? And then what we haven't yet demonstrated, which people have shown previously, is that whether or not PEG-10 is actually an endogenous vector of intercellular RNA gene transfer. We show that we can use PEG-10 efficiently deliver RNA, but we don't actually show that it endogenously is an intracellular vector of gene transfer. And I think that's a really important thing to show if we really want to establish this PEG10. If we want to further establish the role of PEG10 in CNS and human physiology. And then the final question we're really just beginning to ask is, how does PEG10 actually gain tropism, or how does even ARC, for instance, gain tropism? What about it allows it to target specific cells or enter into specific cells? If these proteins are indeed have an extracellular life cycle, what is it about them that allows them to bind to a target cell? Are they functionalized by transmembrane proteins or endogenous fusogens, or we just don't know. And then finally, I just want to say thank you to everybody, including Feng and a graduate student I work with, Blake, and they've been Feng has been the best mentor I could have possibly asked for. He's there for me all the time. So I really just want to say thank you to Feng, and then also thank you to Eugene Koonin for helping us launch this project and doing all the initial genome mining to identify proteins like PEG10. So that's it. And at this point, I guess I'll take any questions.
Thanks a lot, Michael. So for the questions, if you could unmute yourself, ask your question, have discussion, and then mute again so we don't have feedback type issues. Janusz, do you want to ask your couple of questions? The VSVG one you asked before, you talked about CNA, so maybe.
Yeah, I'm very curious because I may have missed this, but how does the capsid-like structure actually end up being surrounded with lipid membrane? So at which point do these two processes, I guess the membrane formation and the capsid formation, how do they intersect in this particular case? And I don't know if you have any thoughts on this, but maybe maybe actually I missed it.
No, that is definitely, we're really interested in like the actual biogenesis of these capsids and how do they actually get released by cells. I mean, at the present, we're thinking that they get released in these multivesicular bodies. They don't bud out like a real retrovirus because they don't actually have the matrix domain, which allows them to anchor themselves on the plasma membrane. But these proteins do not, and so we're not. Yeah.
How do they actually end up in multivesicular bodies? There would have to be some endosomal component to their formation, right?
Yeah. So when we've done co-IP mass spectrometry pull downs on the various capsid proteins, and PEG10 in particular has really strongly associated with like rough ER components, and so we think they're probably trafficked via the rough ER into the Golgi, and then also we get a lot of Golgi components when we do these pull downs. So we think that that's how they're being secreted, but that's about as far as we've gotten so far.
So there's this whole mechanism of membrane sites that contribute to vesicle formation. VAP-A, for example, is involved in this, and so on. So it'd be kind of interesting to see if these intrinsic membrane contact sites participate in this. Can I just have one more question? That sort of,
yeah.
So it's a terrific presentation. Of course, I'm sort of speechless in how interesting this is. But is PEG10 changed, or any of this mechanism is changed in neoplastic states where there's a lot of changes in protein expression and everything else? So do cancers, for example, produce more or less of this stuff, and what would be the consequence of that?
Yeah, so PEG10 has definitely been associated with many different types of cancers, its increase in expression. Whether it's causative or not, I mean, I think that's up for debate. And how would we think about working around that if it is a problem? I mean, I think first we have to actually understand what PEG10 is doing. I think in this initial work, we've definitely hacked it and figured out a nice little delivery vector, but I don't think there's any clear evidence about what PEG10 is actually doing.
If you were to delete PEG10 in cancer cells, what happens?
Well, all I know is that in cancer cell lines we've worked with in vitro, which are HeLa cells, for instance, or even human embryonic stem cells. So anytime we try to delete PEG10, the cells stop dividing, and so we have been unable to actually, we've tried many times to make cell lines out of with PEG10 knockouts, and it's just not possible because the cells just senesce and die. But yeah, I mean, you can imagine, and if we were to actually take this forward as a delivery vector, if we mutate out everything but the capsid protein, and we could maybe produce synthetic particles without an RNA without any of the reverse transcriptase like proteins. You could imagine working around some of the endogenous protein-protein interactions or RNA-protein interactions that PEG10 may have.
Oh, thank you. That's terrific. Very, very interesting. I guess we all stay tuned.
Yeah, please.
Yeah, so adenovirus vector-based gene therapy was burned by, inserting into the genome and causing cancer. So you really need to make sure that this doesn't do that as well. So you said that PEG10 is embryonic lethal, but present all over the place in adults. Is its function known? Is it like involved in mitosis or something? Or
yeah, it's a great question. I mean, there's been a few papers out recently, and including in our hands, where it's associated via co-IP with like different ubiquitin ligases, and there's a paper out from Genentech a few years ago which shows that it directly binds many cellular RNAs, and that it increases their translation, and so PEG10, and we similarly find this, that PEG10 only seems to encapsulate and secrete its own mRNA, but once it's in a cell, it seems to bind with high affinity to many other mRNAs, and especially in the non-coding 5' and 3' UTRs. It really likes to bind highly structured 5' and 3' UTRs, and upon binding, it really likes to jack up their bioavailability. So there's just like a lot more of those mRNAs upon binding. So we've deleted it with AAV. So we've deleted PEG10 with AAV in the developing or the postnatal mouse brain, and just a lot of proteins that a lot of mRNAs that we see it binds are significant, like really downregulated when we delete PEG10. So it seems to bind transcripts and increase their translation or bioavailability, I should say.
Alex, do you want to ask your question?
Yeah, great talk, Alex Milosavljevic from Baylor. It's intriguing that PEG10 perturbation, is critical for cell division. Could the secretion function be specifically abolished to test if secretion itself is critical?
Yeah, that's a really good question. We have not even thought. That's a great point. I think that is. I will bring that up with Fung, and we will talk about that. That's a really good point. One thing we do know is that the reverse transcriptase, we've deleted a number of components from the protein in mouse embryos and tried to see if they develop past E8. And one thing's for sure is that the second reading frame is definitely necessary for its function. So this isn't the secretion question, but there's this unknown role of this second open reading frame. And that for sure is important, is 100% necessary for both mammalian or mouse development, but also probably for the function of PEG10. But
following up on that, the way you figured out that the UTRs are necessary, it looked like the 5' UTR had the highest signal, and then that intergenic, or the sort of in the ORF had the second highest signal. Is that somewhere in between the two ORFs, or could you have a plasmid just with the 5' ORF? Or
yeah, that's a great question. So the part in the middle is actually right at the beginning of the second reading frame, which is very intriguing. So it's right after the first stop codon where there's this -1 ribosomal frame shift. That's where it seems to be that other peak. And yeah, in terms of whether or not you need both the 5' and 3' UTRs, I think we've seen that there's low-level like encapsulation when you just include one or the other, but when you do both, it's just like twice as efficient at least. So we're still like a little bit hand wavy, to be honest, about what are the components of these non-coding sequences. I mean, there's not really like a sequence motif that's that's immediately apparent. Maybe we'll be proven wrong, but we don't see a sequence motif. They're highly structured RNAs, but if you've ever tried folding any sort of RNA, they're all highly structured. So it's hard. We're just trying to figure out, well, what it is exactly about these non-coding sequences which are important.
And
Yeah.
I think you said that one of the cargoes was, I think, 4.5 kb, which is pretty long. Do you, in those EM sort of capsid pictures that you showed, how many copy numbers do you think are within each capsid? Is it one or?
No, this is great. The reason I'm laughing is this has been something we've been racking our heads over for a while. Yeah, I mean, if it's a real retrovirus or retrovirus-like protein, or even a retro element like Ti3 or something, then we would expect there to be 2 copies per particle. We don't expect at present that we're like 100% efficient in promoting trans packaging versus cis packaging of its own mRNA. So we think that it's probably also packaging some of its own mRNA as it forms these capsids. So we're not exactly sure at present. It could be that we are way off and like only a few mRNA molecules are actually getting packaged. It could also be that we're doing fine and we get a lot of mRNAs, and most of them are just 2 copied mRNAs per virus-like particle. But we're still trying to figure out, if you go, I mean, maybe someone here knows better than me, but it seems like the best ways of doing it are pretty rigorous EM studies, trying to figure out how many copies are of a retroviral genome are in a given particle. And we're just now beginning to talk with people who are real experts in that world to try to understand how to do that.
This is really interesting work. Anybody with the final question?
Yeah, this is Travis at UMass Medical School. Great talk. Love your work.
Thank you.
So I guess the big question I always get asked with ARC is, so we've done quite a few biochemical studies, and we can show that ARC monomer, it binds RNA. It binds any RNA we put in front of it. We've seen some of this stuff in the UTR. Maybe there's a sequence motif or a sequence structure that might be conserved. But how do you reconcile that these things are very promiscuous? They'll bind to any RNA in front of it, but yet, as far as we can tell, back to your other question, there's 1 copy of this transcript in 1 fully formed capsid. How does that happen when you have hundreds of these monomers coming together to make a capsid, and then it only grabs 1 copy? So
yeah, great question. I mean, yeah, first of all, it's great to talk to you because you've really inspired much of this work and my postdoc. So thank you. I think we take a little bit of a hint from the line field, the line 1 retro elements. So there's really not much there. It's a combination of sequence motifs that these line elements like to bind, but also it's about just localization of the proteins as they get translated. So as they get translated, they just like to turn around and bind to the closest mRNA that's around. And there's some structural motifs that they like to bind, but really it's a combination of RNA structural motifs and localization. And we think that for PEG10 at least, because we think it's being targeted to the rough ER, we think there's something about these proteins being targeted to the rough ER, and while they're there, there's only a few mRNAs around, and especially its own mRNA, and so it just turns around and binds itself and then gets secreted. And then the ones that aren't, like monomeric ones that are floating around in the cytosol, we think probably have a higher affinity toward, or can bind whatever it comes across. But yeah, we are in the same hand wavy. We don't really know, but that's kind of our working hypothesis at present.
We'll have to leave it there. Thank you so much, Michael, and continued success tracking this down and developing it into a new system.
Great. Thank you. Thank you, Roger. And thank you, everybody, for your great questions and for listening.